Hey, I'm Mr. Williamson, and this video is about meiosis and the formation of sex cells, or sperm making a sperm or egg. Before we get started, make sure you're taking good notes. I think students get the most out of Cornell style because it gives you places to put notations on your notes. But like I like outline, you do whatever you need to do, whatever folds your taco, take good notes. All right? If you've got any questions, make sure you ask them in the Ed Puzzle or ask me in class. Here we go. First of all, kind of a quick review. Remember, the word diploid means 2N. N is equal to the number of pairs of chromosomes in the organism. If we're talking about humans, we have 23 pairs. Therefore, 2N would be a total of 46 chromosomes. All right. Haploid refers to half of that. Hap means half. Is 1N. And 1N, if N is the number of pairs of chromosomes, we have 23 pairs. 1 times 23 is 23. All right. So what haploid cells are? The sex cells, the gametes, the reproductive cells, the sperm or the egg. So when you put a haploid together and another haploid together, you get diploid again. And you really kind of want to make sure you have diploid cells. These are the karyotypes of a haploid, a diploid, and haploid cell. Over on the left over here, you have a diploid cell. Again, what is a karyotype? They take a cell that's going through mitosis or meiosis, and they look for the chromosome count. They pair them up by size, and you end up with the karyotype. Right? So you can see a diploid, 2N, there's 23 pairs, 23 times 2, 46 total. A haploid, although they do not actually make karyotypes of sex cells, sperm or egg, but a haploid would look like the one on the right over here, only has 23 chromosomes. All right. When we replicate them, one of these chromosomes will become one of the chromosomes that we're used to seeing, something like this. All right. Meiosis is the production of sex cells, sperm or egg. All right. And the way it's regulated. The reason I put it in with this unit is because it's very similar to mitosis. Now, the reason that you look real similar to your parents, if you want to know what you're going to look like when you get older, look at your parents. It's genetic. But you don't look exactly, and none of your brothers and sisters, unless you're identical twins, look exactly the same. And there's a reason for that, because you get half of the DNA from your father and half of the DNA from your mother. All right. And because of that, we're mixing that together. So you get some of the genes from your mother, some of the genes from your father. And the result, when we put an N and an N together, is a 2N offspring. Most of the time, when you put N and N together, you end up with a diploid baby. There are cases when that is not always true. And it's usually a mutation or a birth defect. These are 5 examples of genetic birth defects. Trisomy number 21 is Down syndrome. Down syndrome is results in a mental disability. They're also usually associated with vision problems. Teeth that are not, the teeth don't grow in quite right, and like I said, a learning disability. I follow a couple accounts on Instagram of families that have kids with Down's syndrome, and it ranges anything from the severely mentally handicapped all the way to they're really almost fully functioning individuals. They participate in all the things a quote normal individual would participate in. But that's Down syndrome. It's trisomy number 21. Trisomy 18 is Edwards syndrome. Edwards syndrome is usually fatal before birth. So the baby doesn't even come to term. But if it does, they usually don't live more than a year long. They have a really low birth rate. They have clenched hands. They have really small, abnormally shaped heads. And like I said, it's usually fatal. Trisomy number 13, which is Patou, I probably pronounced that wrong, Patou syndrome, is usually fatal within the first year. And again, a lot of these are. And the reason is because there's an excess of genetic information, and the cells don't function well that way. And there's usually some physical defects. Trisomy X is not fatal, oddly, but if you think about it, the X chromosome is what determines, like if you're XX, you're a female. Well, if you're XXX, you're like a super female, but it's not like you're super Wonder Woman. Basically, you're usually taller, and most of the time there are no other issues. If you have a triple X, trisomy X, usually your female features are more pronounced, and I'm not talking about your boobs and stuff. I'm talking about the facial features that make a female, the slenderness of their fingers and things like that. And they're also, like I said, usually taller than normal, or average. I shouldn't say normal, average. Trisomy number 21 usually causes spontaneous abortion or miscarriage, or can lead to death within like weeks of the birth of the baby. But they're all just basically just mistakes in the karyotype. So what you're looking at here is like for trisomy number Down syndrome, trisomy 21. This right here, instead of there only being 2 chromosomes, one from mom, one from dad, somewhere along the line, there's 3. How does that happen? Well, as we go through meiosis here, I'm going to show you how we make the sex cells, and supposedly the chromosomes are supposed to split up, but they don't. So in either mom or in dad, the making of the sperm or the egg, mom passes on or dad passes on 2 of the 21 chromosomes instead of just 1. Or like Edwards, which is trisomy number 18. Again, the mother or father passes on 2 chromosomes instead of only 1, which is normally we split each of these pairs into half and pass on one of the chromosomes, where your partner is going to pass on the other. But if we pass on 2 of them, which would be a genetic mistake, then you're going to end up with the issues. Patou's syndrome is trisomy 13. You pass on 3 of those. The triple X, obviously, this would be XXX. Instead of, well, it wouldn't be XXY, it would be XX. That would be a normal female, and then XXX would be I call them super female, okay, for just lack of another word. And then trisomy 22, again, you pass on mother or father, we don't know, passes on 2 of the chromosomes instead of only 1 of the pairs of chromosomes. All right, that's where the genetic mistake comes in. That's why when we're looking at meiosis, it has to work right, or there can be issues with the baby. So the mixing of the genes, we're going to talk about that. You look like mom and dad, but you don't look like mom and dad, because there's a mixing of mom and dad. But you could have millions of babies, and none of them would actually probably ever be the same. The reason that we actually want the genes to mix, we want some of mom's genes and some of dad's genes to cross over. But think about it, like let's talk about your mother. She's going to pass on one of these chromosomes. Let's just say, for instance, she's going to pass, we're talking about number 9. She could pass on this one, which is from dad, or she could pass on this one, which is from mom. All right. Well, that limits the number of traits that they're going to pass on to you. So in reality, there's actually a mixing of these that goes on, right? And the reason that we want that mixing is because of genetic diversity. We want as much possibility of different combinations of genes and traits as possible. It allows for adaptation, allows for evolution, and it allows different combinations to survive, okay? One of the ways that gets mixed up is the fact that we have 23 pairs of genes. Here's an example where we only have 3 pairs of genes. If we look at all the combinations that we could have, and they're all over here, all the combinations we have. So we have this one, we have of the larger gene, let's call it number 1. There's a combination of a red and a yellow. If we call this gene number 2, or pair number 2, we have a combination of brown and black. And if we call this one pair number 3, there's a combination of blue and green. Well, if we look at each of those, different ways we could combine those, we would come up with 8 different ways. 2 to the 3rd. All right. Give you an example of what I'm talking about. We could take the red one, all right, and combine it with a red one, a brown one, excuse me, and a blue one. So we went all the way across the top right there. All right, or we could take a red one and a brown one and a green one. So we went across the top and then down to that one. Or we could take a red one and a black one and a blue one. So we went like this. Or we could take a red one and a black one and a green one. Sorry. Or we could take a red one, a black one, and a green one. So we went red, black, green. So all the different combinations are over here. These are all the combinations of those three that you can put together. You get to pick one of them from one, you get to pick one from number 2, and you get to pick one from number 3. The way to combine those, there's only 8 combinations. 2 to the third. So it would be 2 times 2 times 2 equals 3, equals 8. 2 times 2 times 2 is equals 8. I can get this. In other words, 2 to the third. All right? 8 different ways. Well, we have 23 pairs. All right? And most of the time, if we're doing this, we're really only counting 22. Well, 2 to the 22nd is 4.2 million almost. Okay? The number is actually right there. But it's 4.2 million. And that's if we don't count the sex chromosomes down in the lower right. If we count the sex chromosomes, then it actually becomes almost 8.5 million different possibilities that we could put all these together. So we could put the first one from number 1 and 2 and 3 and 4 and 5 and 6, etc., and all of them. And then the second one would be all of them, number 1, 2, except for 22. Then we go number 2. And then with the next one, all number 1 and that one except for that one. All the different combinations of the ways to pick one of the pairs of chromosomes from each of these, like 8.5 million if we count the sex chromosomes. All right? But there's even more. There's actually an infinite number of ways to get a different genetic combination passed on to their offspring from one parent. How? When I already showed you, the only way to mix these up is only 8.5 million, roughly. Okay? Well, there's a thing called crossing over. Crossing over occurs during late prophase, early metaphase of meiosis 1. I'll explain that in a minute. Okay? Basically, when they line up in metaphase, you can see where, excuse me, where, let's say this one is from mom and this one is from dad. Okay? By the way, if you don't know what these two symbols mean, all right, this is the female, this is the male. And I probably just gender specified color. Oh well, didn't mean to insult anybody. Hope I didn't. The way I remember it, and I really am serious, is that the female symbol looks like well, the male symbol looks, well, that's how guys are all the time. They're like that, and the female is like, yeah, not down there. You not down. You're not going down. No. Think about it. My point is, you need to remember those because those are symbols I'm going to use. My point is, look, this is crossing over. So these are chromosomes, one from mom, one from dad. Top one's from mom, bottom one's from dad. But the legs of the chromosome actually cross over, and there's an exchange of genetic information. So you can see where this is dad's. It goes right here. But then mom's broke off, and it became part of dad's. And dad's broke off and became part of mom's. The DNA on each chromosome is not lost. It's just exchanged with mom and dad. So while this chromosome right here is mostly, that's a terrible outline. Let's try that again. This chromosome right here is mostly dad. There's some of mom's in there on one of the legs. And this one up here is mostly mom, but there's some of dad's on there on one of the legs. So depending on what part of that the sperm or egg cell gets, there's going to be a common, not only is the mom passing on mostly mom's, but she's passing on a little bit of her dad's also. So there's that 8.5 million combinations from the different combinations of pairs of chromosomes. But inside of that, we can have crossing over taking place. So like, again, working with number 9, I could have the chromosomes like this, but this part here actually belonged to that one, and this part here belonged to that one. So there's if we passed on this, that's the one that's passed on to the offspring, then it would not only contain the information from this one, but it could contain a little bit of the information from the other side also. So the mom or the dad could pass on mostly all of their moms, but a little bit of their dads. Or they could pass on a little bit of most of their dads, just a little bit of their moms. Crossing over makes all of this not only 8.5 million, but an infinite number, because crossing over may not even take place on some of the pairs of chromosomes. Think about it. If you have questions, ask me that puzzle. So let's look at the stages of meiosis. Again, you already know them. You just don't realize it. It's very, very similar to mitosis, but with some variations. That's all. Just a couple things added, or a couple things that don't occur. Okay? So basically, we're starting out with mom and dad. We're going to talk about the formation of sex cells, sperm or egg, and meiosis 1, which is just like mitosis. And then the second part of that is meiosis 2, where each of those split again. The difference is we're not going to replicate the DNA the second time. So let's take a look at the process. Meiosis 1, we go through the same interphase. All right? And think about also, where is this happening? If we're talking about the human body, it's happening in you, in your ovaries or your testes, depending on which one you have. Okay? It's not happening in your arms, it's not happening in your liver. It happens in one place in your body, ovaries or testes. All right? But they still go through all the same interface. There's cells doing their cell thing. First phase, G1, the cell grows. Second phase of interphase, replicate the DNA, S. Third phase, G2, prepare to divide. Same thing so far. Same as mitosis, same thing. That's interphase, not part of mitosis anyways. So then we go into meiosis 1. What happens, let's think about in mitosis, what happens in prophase 1. Okay, what happens in prophase 1 in mitosis? Well, the chromatin thickens up, the DNA thickens up and becomes chromosomes. The nuclear envelope breaks down and the spindle fibers form. That's what happens in prophase here. Prophase 1 in meiosis is the same exact thing as prophase 1 in mitosis, with one little exception. You can see over here that crossing over is starting to take place. That's all. Because the chromosomes, the homologous chromosomes are getting near each other, and there can be a crossing over the legs and an exchange of genetic information. But other than that, in meiosis, prophase 1 is the exact same thing as prophase in mitosis. Same thing. If you know mitosis, you know meiosis. Okay, metaphase. What happens in metaphase during mitosis? The homologous chromosomes line up in the middle. Same thing here. Exactly the same thing. And this is, again, where the crossing over takes place. You can see it taking place here. All right? So crossing over. But all they're doing is lining up in the middle. Metaphase 1. Metaphase. Same. Takes us to anaphase 1. What happens in anaphase mitosis? The chromosomes separate. What happens in anaphase 1 of meiosis? The chromosomes separate. Again, I really like this diagram on the lower right here, because you can see now where I'm using this one as an example, and I'll try to remember to blow this up when I edit it. You can see that there is a red leg to this chromosome, and there's a red and then a partly blue leg. Corresponding over on this side, there is a blue leg and a blue with a partial red leg, where you can see where part of that DNA has crossed over. You've gotten some of the DNA from mom, some of the DNA from dad. If you have anaphase 1, what do you have to have? Probably telophase 1. Okay? Basically, the chromosomes gather in the nucleus, the nuclear envelope shows up, the chromosomes have reached the poles, they might even unravel, usually not. The spindle fibers disappear, cytokinesis begins. The exact same as telophase in mitosis is the same as telophase 1 in meiosis. Well, what we're trying to do, remember, what we're trying to do here is go from a 2N, we replicate the DNA, to 4N, We cut it down in half again to 2N. And now what we have to do is cut it down to 1N. Okay? That's what we're trying to do. So how do we get down? We're at this point right here. We've replicated the DNA and then we split them back. We're back into 2N. Actually, if we did this right, it would be 2N. It would be 2 cells. What we're trying to get it into 2N, and another 1N, and then 1N here, and then 1N here. Okay? So we're trying to get down to 4 1N cells. So how do we do this part? What we need to do is figure out how do we do that. Meiosis 2. First thing it does, prophase 2. The only difference between meiosis 2 and regular mitosis, we don't replicate the DNA because we don't want to replicate the DNA. We want to cut it in half. We don't want to replicate it again. So meiosis 2, we're going to go through mitosis again, essentially, but not replicate the DNA. So we're going to split it all up again. What happens in prophase? Chromosomes thicken up, the nuclear envelope disappears, spindle fibers form. Same thing in meiosis 2. Metaphase 2. Metaphase 2. All right? Here you can see they're lined up in the middle. There's the chromosomes again. Again, with a little bit of moms, a little bit of dads. Okay? Metaphase, they line up in the middle. But the two, because there's 2 cells. Think of it that way, without replicating the DNA. So each of these cells right now is still 2N and 2N. I can make an N. There we go. 2N. Haven't split them up yet. Anaphase 2. Pull them to pull the chromosomes apart to the poles. Still 2N. Anaphase in mitosis. Same thing. Anaphase in meiosis 1. Same thing. It's just pulling apart the chromosomes. Then we get to telophase 2. What happens in telophase? The DNA reaches the poles, the nuclear envelope starts to come back, the spindle fibers disappear, cytokinesis begins. We'll look at the diagram down here. Exact same thing, except there's 2 cells instead of 1 cell, because we didn't replicate the DNA. All right? Each of those cells is different. If we go back here, look at the cell. Well, you can kind of see it. This one is, I guess the best way to do it is this one is red. This one is red-blue, okay? This one is going to be red-blue, but with different one. This one's going to be red. And you can see where each of these cells is a little bit different. Remember, each of these pairs here is 1 chromosome out of that karyotype. Think of it that way. But again, telophase 2, exactly the same as mitosis. All right? When we end up with, you can see where you've got 4 cells that are all genetically different, right? So what we just looked at, and this is not the greatest example, but it brings back the diagram that we kind of I've shown you before. We start with 2N. This would be a normal cell in your testes or ovaries doing its thing. We replicate the DNA so it becomes 4N because we've replicated it, all right? We go through meiosis 1. Prophase 1, metaphase 1, anaphase 1, telophase 1. Splits it into 2 different cells that are now back to 2N. If this was mitosis in your muscle, your liver, your brain, forget the brain cell, whatever, you'd be done. The cells have replicated. But we're not making regular somatic cells. We're not making body cells. We're not making 2N cells. We're making 1N cells. So we go through meiosis 2, we split them again. Prophase, metaphase, anaphase, telophase 2 without replicating the DNA, and we end up with 1N cell. Seeing it in a photograph, you can see the same thing, the phases, not the same thing, the phases. Prophase 1, you start with 1 cell. You have the cells, prophase 1, this would be 4N, not whatever I had there, 4N, still 4N. Metaphase. Still 4N. Now we're down to 2N because we've split it up in telophase 1. We don't replicate the DNA, so each of these is 2N. Not replicating the DNA, we go through prophase, still 2N. This is still, each of these is 2N, and this is the cell, 1 cell here, 1 cell here, 2N, 2N. But now we get down to 1N one N, one N, one N. We've cut it in half. And it should be that every one of those is just a little bit genetically different. Okay? If we're talking about the meiosis in humans, the formation in women, in their ovaries, making of eggs. Here you have, don't worry about the names. It's technically called oogonium. It's going to make, is Latin for egg. You didn't know that, did you? Okay. So we end up with the chromosomes. We replicate the DNA. Okay. So we're 2N here. We're 4N down here. Oh, I can do this. What the heck is that? 4N here. We split them up, and these are going to go back to 2N and 2N. And now we get down to N, 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 and N. But in women, they only produce one egg. So all of these get reabsorbed into the body to produce that one egg that women produce. Now, you got to also remember, in women, it has everything it needs to grow. It has all the organelles, mitochondria, Golgi bodies, endoplasmic reticulum. It has all of those things in the egg because it's a full cell with one N. If we're talking about sperm production in men, again, 2N, 4N, 2N, 2N, and each one of these is N. But the thing about sperm is they're not full cells. They don't have a mitochondria. They don't have Golgi bodies. They don't have endoplasmic reticulum. They don't have the organelles. They have enough energy to allow them to swim. And basically, they're from what I understand, the tails of the sperm they spin, so they that's what propels them. And bam, they're producing 4. So men produce 4 fertilized 4 cells that are 1N, okay, and women produce 1 egg. And the idea is that we get the haploid cell. You can see this is N and this is N. When we get together, this one goes back to 2N, and hopefully you have that normal child, that normal, healthy child. All right? From that cell, it'll go through mitosis. So here we get back to the mitosis part. Wow, what in the world was that? Sorry, guys. Mitosis. It's going to split. That cell is going to split, and it's going to become different parts of the cell. And if you were to take embryology, the study of embryos, early development of embryos, you would learn that you're going to have different cell layers. And one layer is going to be the brain, the nervous system, the dermis, or whatever, the skin, the eyes. Okay, another layer is going to become all these things, muscle cells, blood, things like that. And then you've got another layer that becomes other organs. Once the cell divides, they become different parts of the body, which is why it's important to have all the genetic material for the entire body in this original cell, because when it splits, it's got a code for whatever that particular thing is. Okay? All right, so that is meiosis. Hopefully you took good notes. Real quick review what we talked about. We talked about, again, the little bit of the cell life cycle. We talked about karyotypes and haploid, diploid. We talked about instances of trisomy, where somewhere along the line it doesn't work. Let's go back to trisomy. And I don't know if this is where it happens. I'm just showing you what would happen. Instead of that particular chromosome splitting up and one going that way, one going that way, something goes wrong. And both of them go that way. In other words, it doesn't split. It doesn't do this. This piece right here doesn't go up towards that pole. It gets pulled down with that one. Whatever that cell becomes, if it becomes that one, it now has 2 of those chromosomes for that particular pair of chromosomes. That's what trisomy is. So we talked about trisomy in instances. Again, you don't need to know it. I'm just giving you examples. And then we talked about how we can mix up the DNA. There's 2 ways we mix up the DNA. The combinations of different pairs, and there's like 8.5 million. We have 23 pairs, and all the ways that you could put one of each of those pairs together in combinations is about 8.5 million. But then we can increase that to almost infinity by crossing over. During meiosis, during metaphase 1 in meiosis, there's a crossing over, an exchange of mom and dad's genetic information on that pair of chromosomes. They still have all the DNA, but they exchange it. So now mom's chromosome has a little bit of dad's, and dad's chromosome has a little bit of mom's, and that allows when they split up to increase the amount of genetic variation. All right, and then we talked about meiosis, which is just mitosis, but doing it twice without cell DNA replication. You have prophase 1. The chromosomes thicken up. The nuclear membrane breaks down. Spindle fibers form. You have metaphase 1. They line up in the middle. Anaphase 1. They pull apart. Chromosomes pull apart. And telophase 1. Chromosomes reach the poles. Maybe the cell membrane starts. The nuclear membrane comes back. Telekinesis, cytokinesis. Wow. Cytokinesis occurs. Then you go into the second part where we don't replicate the DNA. And you have prophase 2, thicken up the chromosomes. Metaphase 2, line them up in the middle. Anaphase 2, we split them apart. And telophase 2, where they become 4 individual cells, but they're only one in. I hope you understood all that. If you didn't, make sure you ask questions in Edpuzzle or in the classroom. I hope you took good notes. Good luck.